Students, today we study plant anatomy. Abnormal types of secondary growth, anomalous secondary growth. Anomalous secondary growth and another term abnormal secondary growth is the result of deviation in the Cambian activity from the normal type found in most of the dicots. The abnormal activity gives the abnormal secondary growth, which is known as anomalous secondary growth. Haviland classified anomalous secondary growth into adaptive and non-adaptive anomaly. Adaptive anomaly includes lianas, which are woody climbers of tropics. Non-adaptive type anomaly includes anomaly of Rumex, Boiravia, Bougainvillea, Mirabilis, and Chenopodium. These are the very popular examples of anomalous secondary growth. Types of abnormal secondary growth, anomalous secondary growth. We divide in two types. First one is abnormal behavior of normal cambium. Second is abnormal behavior of abnormal cambium. First we study abnormal behavior of normal cambium. The cambium forms vascular tissues only in the region of vascular bundle. The cambium forms usually large proportion vascular tissues only in the region of vascular bundle. The cambium also forms irregular patches of parenchyma in xylem. Second one is abnormal behavior of abnormal cambium. Formation of rings, vascular abundance embedded in parenchymatous tissue. Formation of rings of vascular abundance embedded in conjunctive tissue. We study first various examples of abnormal behavior of normal cambium. In the first part of our topic, in many woody climbers or lianas, the cambium is normal in position, but its activity is abnormal. It means the normal cambium normal position, but activity is abnormal, and with the result anomalous secondary structures are formed. These types of growth known as abnormal behavior of normal cambium, anomalous secondary growth. We divided in further categories. The first one is the A. Cambium forms vascular tissues only in the region of vascular bundle area. For the example, we study for this region, we study cucurbita stem. The stem of cucurbita, around 10 vascular bundles arranged in two rings of 5 of each. Each vascular bundle is conjoint, open and bicollateral, having an outer and inner cambium. The outer cambium of both inner and outer bundles become active along with the parenchymatous cells of the ground tissue, and they combine to form a more or less wavy ring of cambium. This normal cambium behaves abnormally as it cuts off secondary xylem and phloem in vascular bundle region only, resulting in the increased size of the bundle. Tears of cucurbita stem, as we seen, inner ring of vascular bundle which covered by outer ring of vascular bundle. Each one have five vascular bundles in each ring. These are another diagrams which explain the cucurbita stem. Two rings of vascular bundle. Next one is B, type B. The cambium forms usually large proportion vascular tissues only in the region of vascular bundles. For example, we study bignonia stem. A ring stem shows a wavy outline with prominent wreaths and furrows. At the start of secondary growth, the vascular and intervascular cambium become active and join to form a cambium ring. This cambium ring behaves normally in the beginning, giving more secondary xylem towards inner side and very less secondary phloem on outer side. However, after some time, the cambium rings develops unidirectional areas of cambium at four diagonal points. Hence, at these 4 points, lesser amounts of xylem are cut off internally, whereas massive amount of secondary phloem are cut off externally. These phloem masses intrude inwards, forming four deep wedges of irregular width and supported by transverse bands of sclerotic cells. The TS of bignonia stem. Four wedges sap patches easily seen in center area due to abnormal activity of normal positioned cambium. Cellular TS of bignonia stem. Micrographic TS of bignonia stem. The cambium forms irregular patches of parenchyma in xylem, the third type. For example, we study Urtica dioica. The young stem of Urtica shows a typical dicot structure having a ring of conjoint open and collateral vascular bundles. At an early stage, a normal cambium ring is formed which behave normally, cutting off secondary xylem towards inner side and secondary phloem towards outer side. However, after some time, the cambium begins to behave abnormally. In certain places, cutting off parenchymatous cell on the inner side instead of secondary xylem. After cutting off a group of parenchymatous cells, the cambium again begins to behave normally and resumes its normal activity, forming secondary xylem above the parenchymatous group. This process repeated again and again, resulting in the formation of islands of parenchyma, which are embedded in secondary xylem or wood. These TS easily explain showing alternative bands of secondary xylem and parenchymatous bands.